Hello everyone, I'm Medha. I'm from IIT Madras, Department of Biotechnology. In ISMB 2022, I'm presenting the work CanProSite, predicting lung cancer prone sites using deep neural network. So to introduce about the work, in this work, we try to identify some lung cancer specific disease prone sites, and we use deep neural network for the work. So, lung cancer is a predominant type of cancer which leads to cancer-related mortality worldwide. And the major type of lung cancers are lung adenocarcinoma and lung squamous carcinoma. Those mainly occur due to the higher number of somatic driver mutations in proteins. And if we try to screen these mutations, it will be usually cost and time intensive experiments. So what we tried to identify major proteins associated to lung cancer, and those are KRAS, BRAF, EGFR, ERBB2, and so on. So these proteins belong to cancer sensus genes. Apart from that, we try to identify some specific di and tripeptide motifs, which are specifically preferred in case of disease-prone sites. So those were the another study associated to the work. And if we try to identify the disease-prone sites, it will help to develop targeted therapies which can improve survival rate of patients. So here is one example of the mutation. So in this case, in protein kinase C, this aspartate 475 mutation has been, this site has been mutated to threonine. So what happens? Here you can see the interaction between this aspartic acid with the lysine, whereas when the site has been mutated, the interaction has been lost. So it can create loss of function mutation and mutations, and like there will be several co-occurring driver mutations. So if we try to identify those driver sites where these driver mutations are occurring, that can help to develop some targeted therapies. So to do this, we have developed a server known as CanProSite, and it is available in this link. Now coming towards the material and methods. So we identified 195 cancer sensus genes, which include 4,110 disease-prone sites and 4,139 neutral sites. So neutral sites are basically neutral to the patients. They don't have any impact. And we calculated specifically sequence-based features, so which include physicochemical properties and some di and tripeptide motifs, which were selected from the sequences of proteins. Then we tried to get the evolutionary information using PSSM profiles and conservation scores. We also tried to get some structure-based features such as helix strands in coils, and what is the accessible surface area, and whether the sites are buried and exposed. And those were predicted from sequence. So these are the few of the properties what we calculated. And then we did the feature selection using recursive feature elimination. So in this figure, you can see how we identified the sites and how we calculated the motifs. So here I have given the example of a sequence where in 236 position, tyrosine has been mutated to phenylalanine, cysteine, and serine. And we try to get the di and tripeptide motifs around this site. So we used this information as a feature, and likewise we tried to get the information on specific motifs. So how we developed the model? Initially we loaded the data set, then created the model, we compiled the model, and after that we fit the model into training set and evaluated the model in test set. So the whole data set was split into 90 and 10% ratio, where 90% data was used for training and 10% was used as test. And the deep neural network architecture contains one input layer, hidden layers, and then output layer, which classifies the sites as disease-prone site and neutral sites. So this is the architecture for the deep neural network. Later, moving towards the results and discussion part. So in the figure 4, we have shown the preference of specific amino acid residues towards disease-prone sites and neutral sites. You can see that glycine, aspartate, glutamate, basically negatively charged residues, and also glutamine and tryptophan, they are preferred in disease-prone sites, whereas isoleucine, valine, lysine, and phenylalanine, these are some of the amino acids which are preferred in neutral sites. And this is the ROC plot for the classification model, where you can see that the method had 0.92 AUC in case of training, whereas 0.89 AUC in the case of test set. So this is the overall data for CanProSite. What was the performance on training, tests, and cross-validation? We also tried to compare the method with different existing algorithms, machine learning algorithms, including SVM, AdaBoost, and MLP. And we tried to keep the best threshold using the hyperparameter tuning. And we found that our method is, even in the threshold of 0.5, our method is able to classify disease-prone and neutral sites using with 81%, 81.5% of percent accuracy in tenfold and 79.9 in the test set. So in figure 6, you can see some of the examples of the prediction results where from the proteins which were involved in test set, and this red colored section shows you the percentage of true positives, and you can see that almost in all the proteins our method is able to identify the disease prone sites. So to conclude the work, we tried to identify specific amino acid residues which are preferred in disease prone sites, so which include specifically negatively charged residues and tryptophan. And the developed method is solely based on sequences, and the performance is 80%, which is quite good for a specific kind of method which is specifically designed for lung cancer. And the specific motifs play an important role to discriminate the disease-prone sites from neutral sites, because these peptide-based motifs were found as a key feature in the studies. So, I would like to thank IIT Madras for funding and research facilities, and also I would like to thank Department of Science and Technology for this Indo-Sweden grant. And this is the paper for this work. If you are interested to see in detail, you can go through this paper and you will get more insights for this work. Thank you.